Hi, I'm Poppy and I'm a chemistry student at Imperial College London. I have just undertaken a 10-week studentship at Rothamsted Research where I investigated UK-wide barley yellow dwarf virus substrain variation with Lawrence Branham as my supervisor. I applied for this project because it would give me insight into lots of laboratory techniques that I've not yet had the chance to do before, such as performing PCRs, gel electrophoresis. I also thought it'd be extremely interesting to learn about bioinformatic analysis as well as genome sequencing. This project really stood out to me as plant virology is not something that's only of scientific importance but also of economic value. During my project, I was given many samples of aphids that I tested positive for BYDV by Martin Williamson. The general flow of my research was to pick out the most informative samples. Then we developed primers for BYDV specific cDNA synthesis from the aphid RNA. Then did PCR to amplify up the coat protein region to go on to find a sequencing strategy. We then sent our samples off for Sanger sequencing and performed phylogenetic analysis to see if there was any trends or variation of the strains within the UK. I've really enjoyed working in a big laboratory and a big research facility and really get to grips with this kind of environment. I found it extremely insightful. These projects also taught me how important it is to look at insect resistance and also at strain variation, as this has great effects on the crops and on the environment. Therefore, a career related to plant pathology would definitely be of interest to me. I would also like to say a massive thanks to BSPP for funding this project, as it's really given me an insight into working in a big laboratory and given me lots of new techniques that I will be able to use in the future.